Hello, my name is Sandro Matosevic. I'm an assistant professor in the College of Pharmacy at Purdue University in the United States, and I run a lab developing cell and gene therapies with engineered natural killer cells for immunotherapy of cancer. It is my absolute privilege and honor to be talking to you today during the Laboratories Bioprocessing event during the cell and gene therapy track on our efforts in developing and formulating new immunotherapies with cell-based effectors, specifically natural killer cells for various tumors. So for those of you who don't know what immunotherapy is with cells, cell-based therapy has many different flavors. The most common, the most well-known are perhaps T-cell-based therapies, including CAR T. But beyond CAR T therapies, there are various other immunocell therapies with other immune cells, including dendritic cells, natural killer cells, macrophages, now even neutrophils, and they can take the form of various different types of formulations. This diagram here shows some of the permutations of immune cell therapies in terms of how these therapies can be engineered or formulated to give rise to different T effector cells with different types of specificity. So for example, T cells can be engineered with CARs, chimeric antigen receptors, TCR, T cell receptors, or they can be adoptively transferred into patients just after activation or expansion with cytokines. Similarly, for natural killer cells, they can be engineered with CARs, or they can also be adoptively transferred in certain cases with certain patients for some cancers upon expansion and activation only. Some definitions, a little more specific about these different types of immune therapies. Immunotherapy dates back to 1988 when IL-2 was starting to be administered for various cancers. IL-2 was also the first immunotherapy cytokine that was approved by the FDA for use back in 1996. In terms of immune cell therapy specifically, there are three main types. One is native immune cells that are given to patients just after activation with cytokines. TILs are tumor infiltrating lymphocytes, and these are specifically tumor infiltrating T cells primarily that are cells that reside inside tumors or extracted outside of tumors and expanded and formulated. And finally, there is the third type, which is genetically engineered immune cells, and there are two subsets of those, both TCR, T cell receptors I mentioned before, or CAR, chimeric antigen receptor. And two main types of immune cell therapies that are developed with CARs include CAR T cells and CAR NK cells that have both been clinically and preclinically tested for a variety of cancers to various degrees of success. That the field of immune cell therapy with engineered immune cells, but also native immune cells, is rapidly growing is demonstrated in this slide, where you can see that the rapid number of both increasingly mature cell therapies with especially CAR T cells, but also NK cells, NKT cells, and other types of engineered cells is growing. And this is data that is now 2 to 3 years old, and the number of clinical trials have only increased since then. So there's a lot of effort into really utilizing the immune system as a drug. And these drugs are often called living drugs because they're administered into patients in a formulation that ultimately expands and lives with the recipient for hopefully long term. The mode of action for these drugs is through contact with the target. In this case, the target is a cancer cell. At least it is in the therapies that we're developing in our lab. And so the immune cell has, all immune cells, both T cells, NK cells, and other cells, have various mechanisms that rely on antigen presentation in case of T cells and rely on receptor expression, as in the case of NK cells, within which they encounter the target. So they recognize the target as a foreign entity. They form a synapse upon getting into contact with the target cell, and upon formation of the synapse, the T cell or the NK cell releases proteins, enzymes, and activates various killing mechanisms that eliminates, pierces, and ultimately destroys the cancer cell. And that is how, in an ideal situation, in the tumor, killing would happen. Unfortunately, of course, cell-based therapies don't always act optimally in the tumor. This is why we are developing engineering and formulation approaches to enhance this killing and bring it back to this ideal scenario where these cells can kill a lot of cancer very effectively. One of the ways in which we are trying to enhance the function of these cells by formulating these engineered cell therapies are through the use of chimeric antigen receptors and CARs. I mentioned CARs before. CARs are essentially gene fragments that are engineered to be very specific against antigens presence of cancer on various cancers. And of course, every cancer has a specific type of receptor that hopefully recognize that cancer alone, even though it's not as simple in the human body. But CARs are essentially antigens that, like Velcro, are engineered to recognize and bind to those receptors on cancer. These gene fragments are engineered inside immune cells. They're forced to be expressed by the immune system, by the immune cell, and then they are expressed into proteins, of course, translated on the surface of immune cells. And these immune cells that bear these new CARs are then infused into the patient to perform these recognition and killing activities. These antigens that the CARs are generated against can be either tumor-specific antigens, meaning that they're specific to one tumor, or tumor-associated antigens, meaning an antigen or a protein that is associated with one tumor but is not exclusive to that tumor. They're a lot easier to find, the tumor-associated antigens, but also they're a lot more prone to off-target effects because these antigens are not only present on the tumor, of course. In terms of how do we formulate an immune cell that bears these CARs, these engineered fragments, we have a drug substance, which is really the gene fragment that bears a CAR or any other type of genetic construct that you want to engineer the cell into. And finally, the immune cell, together with the gene, is formulated as the drug product, which is what our ultimate cell therapy is characterized as. And the sources of these immune cells can be, of course, not only in terms of types of cells, such as T cells or NK cells, but they can also be sourced from various tissues. They can be sourced from whole blood, from cord blood. They can be engineered more synthetically through stem cell differentiation. And in certain cases, we have also had success in engineering cell lines. So these are commercial cell lines already available off the shelf into immune effectors through genetic engineering. The attractive part of cell lines is they can, of course, be easily obtained and purchased commercially. The problem with those is that they're more restricted in terms of the ability to use them and the ability and what they can be engineered to target. So when we really talk about the principles of manufacturing and formulating cell-based therapies, we really are looking at a flow through of operations that starts with a drawing of blood from the donor, or in this case, the patient. Blood gets drawn in a typical autologous therapy setting, and this is how the 6-7 CAR T therapies that are on the market in the US are really being formulated. So blood is drawn by the patient to the recipient. The T cells are then extracted from blood in the lab. They're frozen and shipped to the lab where the engineering takes place. Transduction, usually through viral vectors, introduces these CARs into the T cell. The T cells are then expanded for a few days to a couple of weeks to generate doses sufficient for infusion to the patient. After they're expanded, the cells are frozen. They're transported back to the hospital where the patient gets administered these cells. And then this patient gets one infusion of these cells intravenously, and the cells are living drugs at that point and live with the patient for a long time. Of course, there are a lot of challenges at each step of the way, but this is how an autologous therapy where the patient, the recipient, is developed. When we really look at other types of sources of immune cells, we are now thinking about the potential of using immune cell therapy as allogeneic sources, so where you no longer have to rely on the immune system for blood draws, but you can start to develop therapies more of an off-the-shelf, and we'll talk a little bit about that later in the talk. In terms of the differences of these types of therapies, the CAR T therapy that we are engineering is made just in the way that I described recently, just on the previous slide, where the cells are engineered after being drawn from the blood of a recipient. In terms of TIL therapy, on the other hand, these cells are not, there's no engineering per se, but the tumor is excised from the patient, not just blood, and the cells are essentially extracted from the tumor that's excised from the patient. So that's really the main difference between these two types of therapies. And other types of immune therapies have even different manufacturing sort of processes that go through when they get formulated. But this is two examples that I want to illustrate how different therapies have commonalities in terms of how they're made, but also differences in terms of what the specificity of donor sourcing and sort of processing of the tissue. We talked a lot about T cells, and this is because T cell therapies, of course, have enjoyed a lot of regulatory success. We've had, as I said, multiple CAR T therapies approved for use in the US, mostly for blood cancers. But really, one of the cell type that I want to talk about today mostly is NK cells. NK cells have the ability to destroy cancer cells innately. They are similar to T cells, but also different. They also operate on the basis of recognition of foreign cells as targets. The recognition of a cell as a foreign entity triggers the NK cell to form again a synapse with the cell to release perforins and granzymes and other types of proteins that will pierce the membrane of the cancer cell and generate essentially a destruction of the tumor cell. Again, this happens in an ideal situation, this is how it would happen. Of course, in a tumor, a lot of the times we see difficulties in the ability of the NK cell to both kill and destroy the cancer cell. And so we really are faced with challenges in terms of how we can reprogram these cells to better function. The NK cell kills the cancer cell based on the balance of activating inhibitory receptors. So every NK cell has various scenarios in terms of whether it will kill or will not kill the target cell it recognizes. This diagram here shows some of these scenarios. So what's purple, it's where the NK cell does not kill the target cell, and NK cells are illustrated in green in the middle. And the red is when the target cell gets killed. And so the balance of inhibitory activating receptors that any NK cell expresses will dictate whether the cell will activate and kill, or it will be inhibited and not kill. The difference between T cells and NK cells is that NK cells have no antigen presentation required for their killing, meaning they're always on. They scour our body. They get to the site of infected cell very quickly, within a few hours to a few days of infection. Unlike a T cell that takes a lot longer, it requires antigen presentation. So this ability of that NK cell to always be on and always recognize makes it very attractive for development of immunotherapies, because it allows this immunotherapies to be formulated to really leverage the cell's native ability to recognize and eliminate targets without additional immunological mechanisms required to prime the cell. As I mentioned before, when we formulate these cells, we can source them from various sources, and each source has advantages and also challenges and disadvantages. The most common way to source NK cells so far is similar to T cells, is peripheral blood. This is drawing blood from donors or recipients. Other sources include cord blood. Cord blood has a lot more NK cells in it, 30%, so about 2 to 3-fold more than peripheral blood. Cell lines have been quite successful in certain situations for NK cells. There's a cell line called NK92 that has been clinically tested, and it's pretty attractive. Of course, it's a cancerous cell line, so there's a lot of limitations in terms of what it expresses, and it's not quite phenotypically equivalent to a regular NK cell. And of course, induced pluripotent stem cells, we can engineer the stem cells from stem cell sources, NK cells from stem cell sources, and differentiate them into a complete off-the-shelf sources with gene editing. This is a diagram putting all together what I just said. There are 3 main aspects, stages of development of cell therapies. NK cell sources are the first choice that a manufacturer has to make. What type of engineering, gene editing do we want to do to those cells? Do we need to genetically modify them? And how do we do that? What are the targets? How are we going to do it? Is it going to be single target, double edit, or so on and so forth? And finally, treatment. The cell is then formulated into a injectable formulation that gets shipped or frozen for administration into patients. For a long time, a lot of the field really relied on immune cell constructs developed for CAR T cells, but really what we really know right now is that there's a lot of constructs that really NK cell benefit from their own activation patterns. So really the CARs have various components, and what sticks out of the cell, which is the antigen, the scFv, the antibody fragment, the antigen recognition element, is really connected to signaling domains that activate and stimulate the killing. For NK cells, for a long time, as I mentioned, this has been really the same T cell stimulatory domains have been used, but lately more awareness has been placed on really the use of NK-specific activating domains such as CD3zeta, DAP10, DAP12, NKG2D. This slide summarizes some of the main differences in terms of manufacturing, engineering, and treatment and handling of CAR NK versus CAR T cells. What you'll see is that they have a lot of similarities, but I will really highlight just some of the differences. In terms of transduction, so can you engineer these cells? NK cells are particularly hard to engineer genetically. I think T cells are considered a little easier to deal with. That said, NK cells are attractive because they do not participate in GVHD directly, though they can through T cell compartments that they may be infused with. They don't induce cytokine storm, GVHD, and they are always on, meaning they're a little safer as a therapy. And that's one of the main selling points, I think, for a lot of NK therapies, that they are considered more amenable to allogeneic use because of their lower side effects into patients. When we really look at what patients and what cancers are we treating with immune cell therapy, why are we not treating every cancer? I always rely to sort of this graph, which highlights a trend of clinical trials with various CAR T cells for these cancers and these targets that are listed on the x-axis. So, for example, EGFR, HER2, GD2, and so on, which are the antigens present on these cancers. So what you see in green is complete remission, in the peach color is stable disease, and the gray color is no response or progressive disease, meaning a lot of patients treated with CAR T cells with these solid tumors have not responded, and this is sort of the reality nowadays too. If we look at why that is the case, we look at no matter how we engineer the cells, how we treat them, how we engineer them, how we handle the preparation of these cells, if we transfect them or transduce them with lentiretroviruses, if we cell culture them for 2 to 3 weeks, enhance their persistence or their efficiency in terms of dosage, the response rate in solid tumors changes very little, and it's often on average less than 20%, meaning the very few subset of patients benefits from infusion of these engineered cells. And why is the case? We try to understand why these cell therapies have mostly so far benefited patients with blood cancers, and for that reason, all the CAR Ts are currently approved for blood cancers. We found that if we take patient data from brain cancer patients and we look at the association of NK cells with genes that represent inhibitory versus activating proteins and elements of the environment, that we find that NK cells are actively excluded from those tumors that have presence of inhibitory entities in the tumor. So, for example, high expression of CD73 is marked by NT5E, excludes NK cells from those tumors. High expression of CD155, which is marked by PVR, also excludes NK cells from the tumor. On the other hand, high expression of IL-15 or CXCL10, CCL5, which are two chemokines that attract NK cells to the tumor, associates with higher presence of NK cells in those patient samples. So the cancers play an active role in excluding or attracting tumors, something that is really exclusive to some solid tumors versus blood cancers. And that's one of the clues why immune cell therapy, despite various engineering approaches, has not resulted in efficient or consistent responses in patients with solid tumors. There are multiple mechanisms of why the immune cell therapy has really failed to provide responses in solid tumors. So here I'm listed just a few of them. The tumors are heterogeneous. There's a lot of antigen escape. Conditions are unfavorable. There's hypoxia, so low oxygen, low pH. Metabolites float around the tumor and bind NK cells and T cells and then shut them down or inhibit them. There's also overexpression of inhibitory ligands, checkpoint inhibitors, I think everybody's familiar with, PD-1, TIP3, TIGIT, LAG-3, NKG2A. They're overexpressed in the tumor and then shut down the function of immune cells. And also the chemokine, the extracellular matrix and the microenvironment of the tumor is very impermissive to the infiltration, the homing of immune cells. So overall, the tumor microenvironment of solid tumor is very aggressive, and it's very inhibitory to the function of immune cells. And that's part of the reason why just purely engineering immune cells to target one antigen or to sort of just infusing them into the patient in many ways does not provide responses, because there are multiple elements that really work together to fight that infiltration activity of immune cells. One of the mechanisms that we know is very immunosuppressive in solid tumors is CD73, which is an enzyme that is overexpressed in solid tumors, including lung, brain cancer, and many other tumors, where it has been shown to be a negative marker for survival in those patients. It's involved in production of adenosine. Adenosine is a metabolite that floats around solid tumors. It binds the receptors and NK cell and shuts them down. So we don't want adenosine in the tumor. We also know that adenosine in many tumors is pretty negative presence, but we can't take it out from the tumors. We do know that CD73 associates with negative survival in patients. So patients with high CD73 have been shown to survive less. And this is some of our IHC and patient survival data. It also is actively involved in affecting immune cell function. So patients with high CD73 have poorly activated both metabolically and functionally immune cells. And we've published this, and we've done a lot of RNA-seq to show that having high adenosine levels does impair the NK cell's ability to kill the cancer. So what we did to try and target that, we engineered an immune cell that has CD73 antibody blockade combined with an NKG2D CAR. We engineered an NK cell with that and treated a CD73 positive tumor. In this case, we've done both with lung and brain cancer. This is our data with our lung cancer model, where we infused the cells, and we saw some positive effect when CD73 was combined with the CAR-NK cells, but not alone, meaning that there was a synergistic effect of actually eliminating adenosine from binding to NK cells. We then found that these tumors also had higher infiltration of immune cells, meaning that when we blocked CD73, we would liberate more NK cells to enter into the tumor, which is really important. Homing is one of the really big aspects of the tumor. We then took it one step further. We then engineered immune cells to directly target CD73 by engineering a CAR that's made for CD73. And then we showed that when these CARs are lentivirally transduced in the cell can be engineered very effectively to express the CD73 CAR. And then when we treated mice, again, that are bearing these CD73 positive tumors, the tumors would regress, the cell would make more granzyme B, which is a marker of cytotoxicity of NK cells, and would have higher level of homing of CD56 positive NK cells that are expressing these CAR, and that the circulating level of NK cells was not affected by having these genes. However, we also know that targeting one antigen, such as CD73, is not sufficient for a lot of tumors. And while we had really promising results with our cancers, we also found that in a lot of situations, patients have NK cells and that are really not functional. So this is why they cannot fight cancer very effectively. We phenotyped many patients with brain cancer. We extracted their both brain tumors and their blood, and we phenotyped cells both in the brain and in the blood, and we found that these cells are not really very similar to cells from healthy adults. They're very dysfunctional, and that's one of the problems why formulating immune cell therapies with these cells is difficult. Two of the reasons that cancer uses to cause such dysfunction is through both antigens on tumors that the immune cell cannot recognize, and also truly by being actively suppressive, by actually producing, adenosine one example, active immunosuppressants. And this is both of these mechanisms work together to really limit what we can do with immune cell therapies in terms of solid tumors and why the response rates are so low. We also know that a lot of tumors, and this is some of our data with brain cancer, cells are excluded through not being able to just be attracted to penetrate the tumor. Chemokines are involved in complex networks with these tumors, and so we know that a lot of the patients are really not able to get tumors to attract immune cells because of overexpression of this aberrant expression of chemokine receptors that immune cells require. So for example, for our GBM patients, we see that CXCR3 expression is abnormal, which means that the cells are not able to be recruited properly to these brain cancers. And we know that, for example, CXCL10, to prove this, we generated CXCL10 models, and we found that CXCL10 is actually a chemokine that really actively recruits NK cells into the tumor. And so when the tumor has a lot of CXCL10, the NK cells can get there. When we block CXCL10, but whether it's in a mouse model or in vitro, we see that these cells cannot really penetrate the tumor very, very effectively. So to do this, to sort of combine multiple functions and to sort of really target the tumor, effectively developed recently a triple engineered CAR-NK cell that targets two antigens in the cancer, so to avoid antigen escape, and it releases through cleavable peptide linker cleavage CD73 antibody fragment. What it looks like is sort of, this is a 3D rendering of what this therapy looks like. And what we sort of what we did with this therapy is we did a lot of genetic engineering. This is all published data, so I don't have to elaborate on this. We took blood-derived NK cells, we genetically engineered with lentivirus to express this triple CAR. We then combined these cells with the treatment of autophagy. So we used an antibody, a small molecule inhibitor, and genetic inhibition of autophagy in GBM. By eliminating autophagy, we found that these brain cancer tumors can really upregulate the expression of CXCL10 and CCL5, which then in turn recruits more NK cells. So we combined these two elements, and then we treated these brain cancer bearing mice with these autophagy inhibitors. We use chloroquine in this case, and this triple engineered NK cell. And we found that the synergistic effect of these blocking these mechanisms together simultaneously had a tremendous effect on survival and an effect on the efficacy of the immunocell therapy. We think that for a lot of solid tumors, the example of this gives us is that a lot of solid tumors really benefit very greatly from multiple levels. And we then saw also multiple infiltrations of immune cells into these tumors by looking at particularly IHC. We also did a lot of penetration of infiltration studies through our in vitro blood-brain barrier models. Here we have developed here at Purdue to really show that these chemokines are really important in migrating NK cells to the brain. One of our efforts into formulating new cell therapies involves using iPSC, so immune cells, immune pluripotent stem cells, and really it gives us the ability to really no longer rely on blood, really to rely on sort of cells that are able to be engineered completely in the lab. The challenges for using iPSCs really include sort of no control. The control is very poor of the formulated for differentiation and maturation. So in getting the right purity, right quality level of these cells in the right amounts is really still pretty challenging. And it takes a long time. So what we developed is a method of producing iPSC NK cells. It takes about 30 to 40 days, but it generates very pure cells that can expand, and they look like sort of mature NK cells by following this pathway of cytokine maturation of immune cells. So by doing that, we found that these cells are not only very equivalent to immune cells, but they also have all these receptor expressions, and we have this slide shows an example for two cell lines, iPSC cell lines that sort of have receptor expression that follows those NK cells. When we expand and put them in culture and expand them, we get higher purity of these cells. As you see from the right side of the slide, we get a very high CD6 positive and CD negative cells, and on the left side, you see that the receptor expression is essentially equivalent to the ones for both cell lines, which match the ones from the peripheral blood NK cells. To take it one step further, we really engineer these cells to express a brand new triple CAR that has a TIGIT SYNOD receptor. So it's an if-gated construct that allows us to activate the cell specifically in the tumor. So we engineered our iPSCs to express this CAR, and then when we did a lot of characterization and engineering here, you can see this some of our engineering data where we engineered stem cells and differentiated these stem cells straight into mature NK cells. We found that these cells have a very high ability after differentiated into iPSC NK cells to retain this SYNOD CAR that activates TIGIT upon binding to CD155. Upon binding to CD155, the TIGIT CAR releases CD70 antibodies, similarly to what I described earlier, and it has really tremendous ability to treat brain cancer, again, with hugely impactful survival, so highly improved survival and also activity of these immune cells. So really this showed us that using these off-the-shelf immune cells, stem cells, can not only enable us better control of production of these cell therapies, but also really lead to some really exciting engineering approaches where we can formulate these cells to be much more active against cancer. And for the last few minutes of my talk, I really want to just highlight some of the efforts in our lab that are working beyond iPSC NK cells and formulating these therapies that now we have these cells, whether they're blood derived or iPSC derived, into safe delivery, safe immune cells for safer delivery. So I want to highlight here the issue of cryopreservation and sort of what we've been doing to formulate these cells to be given to patients with less side effects potentially, and sort of less damage and sort of less loss upon freezing and thawing. So what we do know that NK cells have to be frozen, just like CAR T cells. They have to be frozen before going into the patient, and the freezing and thawing process damages the cell. And this damage of the cell can happen through slow cooling or rapid cooling. Either of the two can generate either dehydration or ice crystal formation that can destroy the cell upon thawing. We know that this can happen through various mechanisms, and the most common way in which NK cells are frozen is slow freezing. They're frozen very slowly at 0.3 degrees per minute for a period of 2 to 16 hours, and to generate cells that are kept at minus 80 degrees, for example, they get thawed, usually with DMSO. One of the issues that we know with DMSO is that it's very toxic, and we're really kind of interested in understanding what are the implications of infusing patients with high concentration of DMSO, and can we eliminate DMSO so that we can generate cells that are probably safer, less toxic, and without the requirement of washing off the cryoprotectant, so we lose even more cells in the lab. We know that if we remove DMSO, which is one of the main issues to viability of NK cells, we are left with fewer options how we can keep the cells alive. They need cytokines, for example, they need serum, but really don't know what exactly the cells require to really be very functional post-thaw. So we did in our lab, we developed various nanoparticle formulations for DMSO, where we tried to eliminate DMSO and use other cryoprotectants within the nanoparticles to deliver that are protected inside the cell to replace the DMSO that is now no longer there. And so to do this, we wanted to achieve three main aspects. We wanted to either remove, reduce, or replace DMSO. Currently, these are the comparison to the options that we had when we started delivering our nanoparticles. And when we sort of tried to develop nanoparticles to deliver DMSO cryoprotectant to the cell, we sort of had an option of a few things that had been tested so far in the literature, and really mostly focused on cryoprotectants that were outside of the cell. And so we kind of really wanted to go to something that is easy to implement, that has been sort of understood as cryoprotectant. So we picked trehalose, and really for the last two slides, I want to give you some of our data on using trehalose as an intracellular cryoprotectant, and we showed that compared to other cryoprotectants, sort of having cryoprotectants both inside and outside of the cell is beneficial rather than just having cryoprotectant outside the cell, which is the main advantage of DMSO. It can penetrate the cell membrane and sort of act intracellularly very, very efficiently, which is what we cannot achieve with cryoprotectants that are not. So our nanoparticle platform, back to nanoparticle, our efforts really had this form. We had trehalose encapsulated inside nanoparticles. We then delivered nanoparticles to NK cells, and we froze the NK cells, thawed them, and we had some really remarkable results in terms of being able to keep the cells active, very highly cytotoxic, even higher, at least comparable, if not higher than DMSO. We're really excited and by the ability to sort of really gain some insight into what helps these cells. So the bottom line to this is DMSO is very efficient. It penetrates the cell membrane. When we really remove DMSO, we're left with fewer options. Looking at our cryoprotectants that can act both inside or outside of the cell can give us the ability to use safer cryoprotectants without the need to lose cells due to washing or other steps, and to give better infusions of cell therapies to patients. With that, I want to summarize my presentation. I hope I gave you a little bit of a flavor for cell therapy, especially immune cell therapies with engineered NK cells, and how we can leverage synthetic biology, engineering tools, and the various types of formulation, combination therapies with formulations with antibodies, genetic engineering, gene editing, to really generate powerful cellular therapies that can benefit patients that otherwise have very few other options. Thank you very much for listening. Thank you for opportunity to sharing this, and I hope that you enjoy the rest of the event.